डियर फ्रेंड्स, तो आज हम देखेंगे यूनिट 2 बायोटेक्नोलॉजी का। ओके, जो एमसीक्यू ऑनलाइन एग्जाम जो हो रहे हैं उसमें ज़्यादा इंपॉर्टेंट है, तो आप स्टार्टिंग से लेकर के अंत तक देखिएगा, और जो भी प्रीवियस यूनिट है ना वो आपको डिस्क्रिप्शन लिंक में मिल जाएंगे। ओके, फर्स्ट क्वेश्चन नंबर देखते हैं। विच ऑफ द फ्लोइंग स्टेटमेंट इज ट्रू? कौन से इसमें ट्रू स्टेटमेंट है? तो सारे ट्रू हैं। डी ऑल। ए इज अ वेक्टर should have a origin of replication, B is a vector should have selectable marker, and C is a vector should have unique restriction set. The answer is D. कुछ explanation है. A cloning vector is a small piece of DNA that can be stably maintained in an organism and into which a foreign DNA fragment can be inserted for cloning purpose. The answer is D. Next is second question number. Expression vector differ from a cloning vector in having. The answer is D. Control element. Element. The answer is D. Okay. Next is three question number. Combining the DNAs of two species into a single DNA molecules is called recombinant DNA techniques. The answer is B. Recombinant DNA technique explanation is comprises altering genetic material outside an organism to obtain enhanced and desired characteristics in living organism or as their products. The answer is B. Next is four question number. Which of the following does not act as a restriction enzyme? The answer is D. Polydeoxyribonucleotide synthesis. The answer is D. इसका कुछ explanation है. Polydeoxyribonucleotide synthesis is a DNA ligase. क्या है? DNA ligase है. Next is 5 question number. Which of the following enzyme is responsible for making a DNA copy or RNA? The answer is A. Reverse transcriptase. The answer is A. Reverse transcriptase. Explanation is reverse transcriptase generate complementary DNA and form RNA templates. The answer is A. Okay. Next is sixth question number. Which of the following can be termed as a restriction modification system? The answer is A. Restriction endonuclease plus methylase. The answer is A. Next, the explanation is restriction endonuclease plus methylase is termed as a restriction modification system. The answer is A. Okay. Next is 7 question number. Generally, a plasmid vector contains how many elements? The answer is C. 3 elements. Okay. Next is explanation. Cloning site, drug resistant, gene and replication origin are the 3 elements in the plasmid vector. Okay. Next is 8 question number. To make the recombinant plasmid permeable to DNA molecules, which of the chemical is added? कौन सा chemical इसमें added होगा? The answer is A. CaCl2. Okay. Explanation is the cell are treated with CaCl2 to make them permeable to DNA molecules through a process called transformation. The answer is A. Okay. Next is 9 question number. E-core1 is a, the answer is B, restriction endonuclease. E-core1 is a restriction enzyme that cleaves vector at the cloning site. The answer is B. Next is 10 question number. In which stage of genetic engineering a probe is used? The answer is D. Screening. Yeah, screening is किया जाता है. The explanation is in hybridization, which of is one of the method of screening a probe used. Okay. Next is 11 question number. Which of the following enzyme is required for end-to-end joining of DNA? The answer is A. DNA ligase. Okay. Explanation is DNA ligase ligate both blunt and sticky end of the DNA. Okay. Next is 12 question number. Repressor molecules bind to operator. The answer is A. Operator. Explanation is An operator lies partially within the promoter. Repressure bind to the DNA at operator. The expression of gene. Okay, answer is A. Next is 13 question number. Which of the following is required for the preparing the sorry phosphodiester backbone of a DNA during molecules cloning? The answer is D. DNA ligase. Explanation is DNA ligase is required for the preparing the phosphodiester backbone of DNA during molecules cloning. The answer is D. Next is 14 question number. A single strand of nucleic acid target tagged with a radioactive molecules is called. The answer is D. Probe. The explanation is probe are single stranded radio labeled molecules of nucleic acid with a non-sequence. The answer is D. Next is 15 question number. For transformation of microparticles coated with the DNA to be bombarded with gene gun are made up of. The answer is C. Gold or tungsten. Explanation is the metal particle commonly used for the gene gun bombardment include gold, tungsten, palladium and rhodium, platinum and iridium. The answer is C. Okay. Next is 16 question number. The usual source of restriction endonuclease used in gene cloning is bacteria. The answer is B. Bacteria. Explanation is restriction endonuclease represent a class of nucleus originally extracted from the bacterium E. coli. The answer is B. Next is 17 question number. In genetic engineering, the antibiotics are used. The answer is A. As selectable markers. Explanation is antibiotics medium are used to mark or select transformant recombinant bacteria. So we can say antibiotics used as a selectable markers with a marker gene. The answer is A. Next is 18 question number. Which of the following is used to select gene of interest from the genomic library? The answer is D. DNA probes. Explanation is DNA probes are stretches of single stranded DNA used to detect the presence of complementary nucleic acid sequence, target sequence by the hybridization. Okay, the answer is D. Next is 19 question number. The enzyme which are absolutely necessary recombinant DNA technology are. The answer is C. Restriction of endonuclease are and ligase. The answer is C. Okay. Next is 20 question number. Which of the following is a plasmid? कौन सा इसमें है plasmid? The answer is A. PBr322. Its explanation is PBr322 is a plasmid and was one of the first widely used E. coli cloning vector. Created in 1977 in the laboratory of Herbert Boyer at the University of California and San Francisco. The answer is A. Okay. Next is 21 question number. The TI plasmid is used for introduction gene into. The answer is B. Plant cell. Explanation is in plant genetic engineering the TI plasmid can be used carry foreign gene into the plant cells. The answer is B. Next is 22 question number. Which technique is used to separate protein fragments based on size? The answer is B. Southern blot. The answer is C. Aggregate gel electrophoresis. B is not right. C is right. Aggregate gel electrophoresis. Explanation: electrophoresis is laboratory techniques used to separate DNA, RNA or proteins molecules based on their size and electrical charge. Okay, answer is B. Next is 23 question number. Restriction endonuclease A cut the DNA molecules at specific sites. Explanation is restriction endonuclease cut the DNA molecules at a specific site. It is also called molecule season. The answer is A. Okay. Next is 24 question number. Polymerase chain reaction technology PCR it is used for the DNA amplification is the answer is true. Okay, true. Next is 25 question number. The plant obtained by gel electrophoresis is during separation and sustained by C. Ethidium bromide. Okay. Explanation is ethidium bromide, the short form is ETBR, is used to stain DNA in agarose gel electrophoresis, but this dye is mutagenic and carcinogenic. Okay, answer is C. Next is 26 question number. Which of the following is not the cloning vector? कौन से cloning vector नहीं है इसमें? The answer is C. Neurospora. Neurospora. The answer is C. Next is question number. Construction of recombinant DNA involves. The answer is D. Cleaving DNA segment with endonuclease and rejoining them with ligase. The answer is D. Okay. Next is g-patrevision classes को सब्सक्राइब करें, शेयर करें एंड लाइक एंड कमेंट जरूर करें. ओके, तब तक के लिए बाय. नेक्स्ट वीडियो मिलते हैं.